Welcome back to Virology. This is lecture number 3, Genomes and Genetics. What is genomes and genetics? Well, the viral genome consists of DNA or RNA. And I want to talk about the different configurations of DNA and RNA, how they work, and then genetics part is how we can study them. Now, in the 1950s, there was a virology breakthrough, very important. 2 sets of experiments showing that the viral nucleic acid is the genetic code. Now, today, obviously, we take that for granted, as we should, but it wasn't so well accepted at the time. There were 2 experiments, one with the DNA bacteriophage and a second with tobacco mosaic virus and RNA virus. So the DNA experiment, called the Hershey-Chase experiment, which probably many of you have seen before, involved labeling bacteriophages either with sulfur, radioactive sulfur to label the proteins, or with radioactive phosphorus to label the DNA. And by label, we mean the radioactivity is incorporated into the nucleic acid or the protein, and then you can easily trace it. And so Hershey and Chase took those 2 different kinds of labeled viruses, allowed them to attach to E. coli, these were bacteriophages, allowed them to attach to E. coli for just 5 minutes so the genome would get in, and then they put them in a kitchen blender to shear off the phages so no more would get in, keep the noise down. If you ever go to Cold Spring Harbor, there's a library there with one of the blenders that they used. Okay, so then after they shear off the phage, either the protein or the nucleic acid is in the cell, they let the new phages develop and they measure where the radioactivity is. And there's no radioactivity in the protein of the next generation of phages, but there is radioactivity in the DNA, showing that the DNA is the genetic material. Luckily, they used a phage that just injects its DNA into cells, because many phages, the whole thing gets into cells. That would have been confusing, right? Both protein and P32 labeled nucleic acid would have gotten in. The next experiment with tobacco mosaic virus is shown here. We don't study this in high school, but I think it's pretty cool. It's called the Frankel-Conrad experiment. Frankel and Conrad identified 2 strains of tobacco mosaic virus. We'll call them green and red. And they could distinguish the coat protein of these viruses readily on gels. And so the coat protein, of course, is what makes up the virus. It coats the RNA in many, many copies. And we'll talk about structure maybe next time, yeah. And what they had found out is that they could separate the coat protein from the RNA and put them back together again, and they would make infectious virus. So they separated the green and the red coat and RNA, and they co-mixed them. So they took green RNA and mixed it with red coat protein, and red RNA and mixed it with green coat protein. They took these reconstituted particles, infected leaves, tobacco leaves, with them, and the virus that came out, the coat protein always matched the RNA. So the green RNA always gave rise to green coat protein, and the red always gave rise to red coat protein. So again, the genetic information of the RNA virus is in the genome. So 2 classic experiments. The bigger surprise, though, as people began to study viruses, even though there are thousands and billions, actually billions and billions of viruses on the planet, there are only 7 different kinds of viral genome. And you can remember that because that's the number 7 train. 7 different kinds of viral genomes, which is amazing. And another thing that will help you a lot is that viral genomes have to make mRNA that can be read by host ribosomes, because remember, the virus cannot translate protein. It doesn't have a protein synthesizing machinery, so it must make mRNA that can be recognized by the host cell. Every virus follows this rule. There's no exception. So those two things, 7 genomes in mRNA, were used by David Baltimore, a Nobel laureate, to organize these 7 genomes in a simple way. So he actually worked with 6 genomes, because at the time of this, in the early 70s, one of the genomes had not yet been discovered. So what he did was to place mRNA at the middle and then put all the different genome types around it and trace how you get to mRNA from those genomes. So here's mRNA, and so some genomes are double-stranded DNA. This can be transcribed directly to make mRNA, so it's a direct step from double-stranded DNA to mRNA. In fact, among the DNAs, only double-stranded DNA can be transcribed to form mRNA. It's something you should remember. Single-stranded DNA cannot, gapped DNA cannot. So there are 2 other kinds of DNA, virus DNA genomes. There's single-stranded and viruses that are partially double-stranded. They're gapped. They can't be transcribed. They would first have to be repaired to make double-stranded DNA, and then they can be transcribed to make mRNA. Then we have RNA viruses. We have double-stranded RNA viruses. So the double-stranded RNA viruses have a plus strand and a minus strand, right? But the ribosomes can't access the plus strand. It's locked into this double strand form. So the virus has to first make mRNA from that double strand. We also have negative strand RNAs. Now, negative strand, the definition of negative strand is the opposite polarity of message. So these virus genomes cannot be translated into protein. So they first have to be copied to plus RNA. We also have viruses with plus RNA, which means it's the same polarity as message. And there are 2 classes of viruses. Those ones down here that go through a negative strand intermediate. But this genome, in fact, in theory, could be translated to make protein. And then the other kind of RNA virus goes through a DNA intermediate. That's the retroviruses. And the DNA becomes double-stranded, and from there, the mRNA is made. So this was a very brilliant way to categorize the genomes. And again, every virus on the planet seems to fit into these 7 classes. We haven't found any other ones. Not sure why, but that's what we know. So before we go any further, let's make some definitions, because there are words that are used in different ways. So the plus strand of mRNA is, well, the mRNA is defined as the plus strand. It's ribosome ready. It can be translated. And plus doesn't mean anything in terms of electricity or polarity. It's just a convention, right? And in a DNA, the equivalent polarity is also the plus strand. So the same polarity as message in DNA is plus strand. And the complements are called negative strands, just to show that they are complementary. And another thing you should remember is that not all plus RNA is actually messenger RNA. It's not always translated. We'll see some examples of that today. So the elegance of this Baltimore system, I think this is a brilliant classification scheme. You only need to know the nature of the genome, single-stranded RNA, gapped double-stranded DNA, and you can tell me the steps that you need to get to mRNA. It's really brilliant. So again, 7 classes of viral genomes, double-stranded DNA, gapped double-stranded DNA. single-stranded DNA. So those are the three categories of DNA. And then double-stranded RNA and single-stranded RNA of plus and minus polarity. And one of those plus single-stranded RNA has a DNA intermediate. So why is mRNA placed at the center of the Baltimore scheme? A, because all virus particles contain mRNA. There's no specific reason. Because all viral genomes are mRNAs. Because mRNA must be made from all genomes. Because Baltimore studied mRNA. Let's see how we did. Okay, because mRNA must be made from all viral genomes. That's why he put it at the center. All virus particles do not contain mRNA. There's no specific reason. Nobody got that. That's great. All viral genomes are mRNAs. Nobody got that. That's great. Because Baltimore studied mRNA. Even if he did, it wouldn't be the reason why he put it at the center. It would have to be a logical reason, right? Good. Now, in addition to these 7 classes of nucleic acid, the genomes are structurally diverse. So double-stranded DNA can be linear, it can be circular, and let's see if we go down there. Double-stranded, it's linear as well. If you go further down, there's a double-stranded linear version where the ends are covalently linked. And then we have RNAs that can be circular, they can be segmented, they can be plus or minus, of course. They can be double-stranded. They can have proteins attached to the ends. And here's the weird DNA of hepatitis B viruses. These are the viruses that RFK Jr. thinks we should not be immunizing kids against. As if he would know if he read any papers, but in fact, if you're infected in the first 5 years of life with Hep B, you will be infected for the rest of your life, you won't clear the infection, and you're likely to develop hepatocellular carcinoma. If you're infected as an adult, you can clear the infection, but not as a kid. That's why we immunize newborns. Okay, so this is Hep B. Genome is gapped. It's partially double-stranded. It's got a piece of RNA on it. It's got a protein on it. So it's very weird, and it has to be repaired before it can be transcribed. Anyway, the point here is that they come in a lot of configurations. So what's the function of this? What's the function of having this genome diversity? Why isn't there just DNA? Why do viruses have both DNA and RNA-based genomes? Well, we think, remember, there was an RNA world at one time on Earth. So the very first molecules that evolved were likely RNAs, which remarkably could self-replicate in the early organic Earth. And they got to considerable length, and a whole RNA world developed which had catalytic activity. Like those RNAs had the ability not just to replicate, that's catalytic, but also to cut themselves, self-splicing. And eventually, DNA evolved. How that happened, we can talk about later. It's very interesting. But we think that the viruses with RNA genomes are relics of the RNA world, basically. That's why they exist, because they were once RNA, and probably the first viruses were RNA viruses as well. And also today, we have on the planet molecules called viroids, which are probably direct relics of the RNA world. They're very interesting. That molecule up there is a viroid. It's a circular RNA, highly base paired, as you can see. It doesn't encode any protein. And yet it can, well, it probably infects many kinds of organisms. We thought they only infected plants, but in the past year, we found them all over the place. They're probably copied by host enzymes. And they cause disease in plants. They're agriculturally really important, but they don't encode any protein, which is really interesting. These are probably examples of the kinds of molecules that existed in the RNA world. So that's why there's RNA and DNA. But As for linear, circular, segmented, double-stranded, single-stranded, plus and minus, why do we have this diversity? We don't have an answer, but my feeling is they're all evolutionarily competitive. They work. They all have their advantages, and therefore they can compete with one another. If I were designing viruses, I would make all viruses plus-stranded RNA viruses, because the RNA is mRNA. As soon as it gets in the cell, it can start replicating. It's very quick, and it's a simple reproductive style. But they're not all RNA viruses. They're not all plus RNA viruses. So years ago in this course, before it was open book, I used to say memorize the 7 genome types. You don't have to memorize them, but you have a copy. Know where your copy of this chart is for the exam, and it'll help you. So if I tell you plus single-stranded DNA, how do you make proteins? You'll be able to trace that. In fact, in the old days, students used to make cheat sheets, right? They're some of the most creative things I've ever seen. You can do a lot on a computer. It's really fine. But some people drew them in all different colors with the tiniest handwriting. Oh my gosh, it was so creative. I miss those days because you don't need to do it anymore. So what's encoded in a genome? Lots of things, but just some examples. Gene products and regulatory signals for protein synthesis. Mainly the giant viruses have these. We'll talk about that. Proteins to help replicate the viral genome. Obviously, polymerases are an example, but other proteins as well. Proteins involved in assembly and packaging of the genome. Proteins involved in the regulation and timing of the replication cycle. Modulation of host defenses, that's an important one. Our host defenses are really good. In a healthy human, a young, healthy human, host defenses are really good, and they would eliminate every virus infection if viruses didn't have countermeasures, which they all do. And finally, proteins that allow viruses to spread to other cells and other hosts. Now, this is an interesting giant virus. Remember, I told you the other day that not too long ago, we discovered really, really big viruses. They're called giant viruses. And this one is cool. It's called Tupon virus. And that's an electron micrograph of it. Of course, it's colorized, but it's very pretty, I think. This one encodes 20 amino acyl tRNA synthetases, right? The enzymes that couple specific amino acids to tRNAs. It encodes 70 tRNAs, multiple translation initiation and elongation proteins, multiple translation related genes like for tRNA and mRNA maturation. Basically, it encodes the most complete translational apparatus of the virus sphere. The only thing that is not encoded is the ribosome. It's got everything else, but no ribosome. So why do we have these extra proteins? Probably to modify the cellular translation apparatus to favor the production of viral proteins. And there's some evidence, there's some experimental evidence for that as well. Now, things that are not contained in viral genomes, this list gets shorter every year as we discover genes that are in the genomes. We haven't found any genes encoding the complete protein synthesis machinery, right? The Tupon virus has everything but the ribosome. I used to have on this list no genes encoding proteins involved in membrane biosynthesis, but recently we found these in some giant viruses. No classical centromeres or telomeres that you would find in a standard host chromosome like we have. But we probably haven't found them yet. 90% of giant virus genes are novel, so there may be genes encoding telomeres or centromeres. Who knows, maybe a virus someday will be found that encodes a ribosome. But it's a lot of energy to go through to encode a ribosome when you can just steal it from the host cell. So I wanted to show you the biggest and the smallest known viral genomes, just to give you an example of the span. So these are the top 10 viral genomes. So the biggest one is Pandoravirus salinus, or Salinus, 2.47380, 2.4 million base pairs of double-stranded DNA, and encoding 2,500 proteins. It's huge. The next one down is Pandoravirus dulcis, 1.9 million base pairs of DNA, 1,400 base pairs. And there's Tupon virus that encodes all those translation proteins. And then many other viruses down to 600,000. So I want to point out on this graph here, Haemophilus. This is a bacterium with a genome of 1.8 million base pairs. And so you can see two viruses have bigger genomes. So these are really big. And as I said, 90% of the genes, we have no idea what they encode. You know, we can look at some and say, oh, this is an enzyme of some sort. But 90% we have no idea. And maybe there are useful genes in there, and a lot of people are working on that. How about RNA? So these don't even come close. The biggest ones are, well, these are all nidoviruses. Nidovirus is the order of viruses that contains the coronaviruses. And so there are 3 viruses with 41, 35, and 33 kilobases of single-stranded RNA that infect planaria and aplysia and pythons. And then coronavirus genomes typically are 30 kb. The SARS-CoV-2 genome is about 30 kb in length. But most RNA virus genomes are around 10 to 12 kb in length. They don't get very big. And we'll talk about reasons for that later. But in general, RNA is very fragile compared to DNA, and it can't get very big. How about smallest known viral genomes? So we have at the very top what are called satellites. These are RNAs that are packaged into capsids that are provided by a helper virus. So they're helper-dependent viruses. The genome is very short. You can see 220 base pairs. And this one encodes no protein. It's completely dependent on a helper virus to get encapsulated. But they do get encapsulated, and they can infect cells. Then under that is hepatitis delta RNA, which is a human pathogen. This encodes one protein, and it depends on hepatitis B virus to assemble a virus particle, a delta particle. And so you only find delta in people who already have hepatitis B virus infection. And we think that hepatitis delta makes hepatitis B virus infection worse. Then we have some DNA viruses thrown in here, circoviruses, anelloviruses. These are viruses with circular single-stranded DNA. Everybody seems to be infected with these. They encode just a few proteins. They don't seem to cause any disease in anyone. So when you give blood, it's not tested for circo and anello because everyone has it. And you wouldn't have a blood supply if you rejected it on that basis. There's hepatitis B virus DNA. It's pretty small, only 7 proteins, and then a couple of other RNA viruses. So you can see the big difference in size between the biggest and smallest. Yet, to me, what's really interesting is they all work. They're all effective viruses. They can get into cells, take over, and make lots of new viruses. So why this range in size? Well, why isn't the right word. What's the function? What do you get by having a bigger genome? Well, we really don't know. Next question is, what information may be encoded in a viral genome? A, complete protein synthesis systems. B, centromeres or telomeres. C, enzymes to replicate the viral genome. Or D, all of the above. So which may be encoded in a genome? All right, let's see how we did. A little bit of a mixed bag, but the answer is enzymes to replicate the viral genome. complete protein synthesis systems. No, we don't have ribosomes encoded in the genome, right? And not centromeres or telomeres. That was on one of the slides. So a lot of you picked all of the above, but it's just C. Let's talk a bit about viral DNA genomes. Pretty much, most of the viruses of bacteria are double-stranded DNA viruses. And of course, there are lots of eukaryotic DNA viruses as well. And in general, they emulate the host. The host is double-stranded DNA. So a lot of the processes that occur in a virus-infected cell are similar to what happens in an uninfected cell. But almost all viral genomes are not like cell chromosomes. Now, as you know, cell chromosomes are what we call chromatinized, right? The DNA is wrapped around histones, and that's how you get it all compacted into the nucleus, and it's also a mechanism for transcriptional regulation. But most viral genomes do not look like this in the virus particle. And viral DNAs do some different things compared to that of the host, which we'll see as we talk about DNA viruses in more detail. But I wanted to show you some DNA virus genomes as examples. So here are two classes of DNA, double-stranded DNA virus genomes. So remember, in the Baltimore scheme, this double-stranded DNA can be transcribed into mRNA, and you can make protein from that and more virus particles. One class of these double-stranded DNA genomes is shown on the left. They're relatively small. Like here's a circular double-stranded DNA virus, about 5,000 bases in length. It's called polyomaviruses, and we'll talk about those later. These are too small to encode the DNA polymerase and all the stuff you need to copy DNA, so they are copied by host DNA polymerase. Whereas the genomes on the right are bigger. They encode their own DNA polymerase, which gives you some advantage. You can do more regulation of the timing of DNA synthesis. And these include adenoviruses, 30 to 50 kilobase pairs of double-stranded linear DNA, herpes viruses, double-stranded linear but much bigger, and pox viruses, 130 to 375. Monkeypox virus, of course, the cause of mpox is a member of the Poxviridae. And here are some DNA viruses that we'll be talking about in this course. We have a few viruses that we focus on. So adenoviruses, which we've talked about the genome, and herpes viruses and pox viruses. Those are the DNA genomes that can encode their own polymerase. And then polyomaviruses and papillomavirus have the small genomes, about 5 kb circular DNA that need the host for DNA replication. These are human pathogens, all of them. And that's pithovirus on the right, one of the giant viruses. It's not drawn to scale. It would not fit on the slide if I drew it to scale. And that blue thing in the middle is the DNA genome, and then the virus encasing it is shown there in green. And those little yellow bits at the bottom, that's called the cork. You know, these were discovered by the French, who like corks in general, and they like pulling corks out of wine bottles, right? And so they say this is the cork. The cork comes out and the DNA then follows. And the hepatitis B viruses have this funny gapped DNA. So here on the left, lower left, that's the genome. It's circular, but it's not fully double-stranded. There's a single-stranded region, plus there's a piece of RNA on it, and there's a protein. That genome can't be copied to mRNA. The only thing that can be transcribed to mRNA is double-stranded DNA. We got to remember that. It's really important. So it has to be repaired. And what's interesting is when this molecule enters the host cell, you know, any DNA that enters the host cell if it's damaged, the cell will fix it. It doesn't like to have single-stranded DNA in the nucleus or broken DNA. It will repair it. It does it a favor. So it's the host that takes off the RNA and the protein and fills in that gap. And then it's fully double-stranded. It can be transcribed, and boom, the reproductive cycle begins. So what happens is these genomes get in the cell, they're repaired, and then they can be copied into RNA. Some of it is mRNA, which made into proteins, but a subset of that RNA is copied into double-stranded DNA. And that's what we find in the virus particle by an enzyme called reverse transcriptase. We'll talk about that in more detail and the reason for that. Single-stranded DNA genomes. Lots of viruses look like this. We have ones with circular single-stranded DNA, like these circoviridae that I say infect all of us. One of them is called TT virus. It's, again, a ubiquitous human virus of apparently no consequence. And then we have the parvoviridae, one of which causes a childhood rash disease called fifth disease. And there are parvoviruses of cats and dogs, which can be lethal. So if you have such a pet, you should vaccinate them because they will catch it from other dogs. So these viruses can package both plus or minus DNA. It doesn't matter because in both cases they have to be repaired by the cell to make double-stranded DNA. Again, the cell sees this single-stranded DNA coming into the nucleus and then goes, nope, you're not coming in here, so that repairs it. And that makes it double-stranded, which can then be transcribed, and then you can get protein and DNA synthesis and so forth. Which DNA genome on entry into the cell can be immediately copied into mRNA? Double-stranded DNA, gapped double-stranded DNA, circular single-stranded DNA, linear single-stranded DNA, all of the above. Okay, how did we do? So the answer is double-stranded DNA. Most of you got that, right? That's the one thing you have to remember. It's the only molecule that can be directly transcribed into mRNA. All the others have to be repaired first. RNA genomes. These seem to dominate the eukaryotic virosphere. In other words, all the viruses of eukaryotes. We think that there are not many in bacteria. So here's a chart showing that. This is plus RNA, double-stranded RNA, and minus RNA. And these are the numbers of families in plants, invertebrates, bacteria, archaea. And you can see the bacteria, there are very few of RNA viruses of any kind. I think this is a sampling artifact. People are now discovering these RNA viruses in bacteria, so this may change. But RNA viruses can be in many configurations. They can be plus or minus, or they can be segmented or double-stranded or even circular. The key to understanding RNA viruses is that mammalian cells have no enzyme to copy them. They have no RNA-dependent RNA polymerase, which you would need to copy an RNA into another RNA. So every RNA virus on the planet has to encode its own polymerase, with the exception of those satellites that don't encode anything, of course. They depend on a helper virus. And so these polymerases, which we'll call RDRPs, they produce RNA genomes and mRNAs from RNA templates. So let's have a look at some of these viruses. So rotaviruses, these are causative agents of human gastroenteritis, vomiting and diarrheal disease, very common. Used to be a big killer of babies because they get dehydrated and it's very hard to keep a baby hydrated. They have double-stranded RNA genomes which occur in segments. You can see here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 segments. And these double-stranded RNAs, they have a plus strand in them, but the ribosome can't bind to that and translate it. So first, an RNA polymerase in the particle has to make mRNA from that double-stranded RNA, and then that can go on to make protein and new virus particles. So another thing to remember, the plus RNA in a double-stranded RNA molecule cannot be accessed by ribosomes. Then we have plus-stranded RNA viruses, which I think are the most simplest that you could devise, but they're not alone. Plus-stranded genome, which can be directly translated into protein once it gets into a cell and start the replication cycle. And here are some examples of RNA viruses of plus polarity. Coronaviruses have the biggest RNAs. Flaviviruses like yellow fever virus, West Nile, Zika, slightly smaller. Picornaviruses like poliovirus and togaviruses, all plus-stranded RNA viruses of significant pathogenic potential in people. And so here are the actual viruses, the Picornaviridae. You may be hearing of outbreaks of norovirus lately, also called winter vomiting disease, because it tends to happen in the winter. This is also the virus that causes outbreaks on cruise ships and sends them back into port. It's a small plus-stranded RNA virus. There you have your coronaviruses, which include not only SARS-CoV-2, but MERS, SARS, and others. Flaviviridae, yellow fever, West Nile, Hep C, and Zika, and togaviruses, including rubella and equine encephalitis virus. So equine encephalitis virus is a mosquito-borne virus that can cause illness in horses and also in people if the mosquito should bite them. And then plus-stranded RNA viruses with a DNA intermediate. And there's one viral family here, Retroviridae, and there's two human pathogens, HIV 1 and 2, and HTLV. There are multiple HTLVs. And these are fascinating viruses because the viruses contain a plus-stranded RNA, yet when the viruses infect cells, that RNA is copied into a single-stranded DNA and then to a double-stranded DNA, which integrates into the host, and then there it can be transcribed into mRNA to make protein. And so these are all obligatory steps, the synthesis of DNA, double-stranded DNA, and integration into the host. So the genomes of these retroviruses are shown here. They're basically mRNAs produced by the integrated DNA. I'm going to talk more about these later. They're very interesting. Minus-strand RNA viruses. These come in different configurations. They can be segmented, like influenza viruses have 8 single-stranded RNA segments in their virus particle, as diagrammed up there. And some have non-segmented genomes, like measles virus or rabies virus. Now, as a minus-strand RNA, it cannot be translated, right? The ribosomes will not be able to make protein from it. So when this enters a cell, it has to be copied to an mRNA. And the only way that can happen is for the virus particle to contain an RNA polymerase, because cells do not have RNA polymerases that can copy these negative strand RNAs. So every negative strand virus needs to carry within the particle an RNA polymerase. Now, all RNA viruses encode an RNA polymerase, but only the minus-strand viruses have to have the polymerase in the particle. The plus-strand viruses don't, because when that RNA gets into cells, it's immediately translated to start the infectious cycle. It's very cool. So here are some examples of minus-sense RNA viruses. We have the paramyxoviruses, measles, and mumps virus. These are enveloped. In fact, these are all enveloped viruses. Rabies virus, the filoviruses, Ebola virus and Marburg virus, very large filamentous RNA viruses, orthomyxoviruses, influenza virus, and arenavirus. So Lassa virus was first discovered in Nigeria in the 1960s. It was the first emerging infection discovered where the virus is endemic in rodents and it started infecting people. It was the first example of a zoonotic infection. And the history of that is told very nicely in this book, Fever by John Fuller. Great story. And part of it takes place at the Columbia University Medical Center where they brought one of the patients. Anyway, I graduated college not knowing what to do with my life. And I read this book and I said, viruses? They can do this? And so that made me want to be a virologist. So that book is the reason. It's really a good book. And I think you can get it free on Amazon. So you should check it out. It's a very quick read. Now, viruses that have segmented genomes in pieces, they can do something that no other genome can do. They can do reassortment. What is that? So here are two influenza viruses with 8 segments each, and one of them has red and one of them has blue segments. When these two viruses infect a cell and the segments reproduce, they mix up, right? And so the new viruses that are produced can have a mixture of segments. That's called reassortment. So you can see this virus here on the right has 1 red out of the 8 RNA segments. This is very powerful for genetic diversity and is why pandemic influenza viruses are generated. So right now, we're worried about H5N1 influenza viruses causing a pandemic. And the reason we're worried is because they're infecting so many different animals now. We're worried that they may reassort with another influenza virus infecting an animal and acquire transmission potential in people. So right now they don't transmit, but we're worried that reassortment can confer that. So what do? You appoint someone to head HHS who wants to cancel infectious disease research. That's the smart thing to do, right? There are some genomes called Ambisense, very interesting, because their RNAs have a little bit of plus and a little bit of minus polarity. And Lassa virus is an example of that. It has 3 RNAs, single-stranded RNAs with a little bit of plus and a little bit of minus, as you can see here. They're officially negative strand viruses because there is an RNA polymerase in the virus particle. And that is because when these RNAs infect cells, not enough can be translated to get the infection going. So it has to be copied by an RNA polymerase. Now, RNA, I've been showing RNAs as lines like this, squiggly lines. But that's not how they look, of course. They're highly structured, and the structure is relevant. So here in panel B is the structure of a plant RNA virus. And you can see these, what we call stem loops. So the RNAs can base pair with each other in local regions and form these double-stranded structures. And they can be highly complicated, as you can see here, highly extended. And not only that, there can be long-range interactions. So this red region from here and here are base pairing, and the same with the blue. So structured RNAs are really important. On the right is the 5' leader of HIV-1, which has a number of stem loops. And this is, below it is the actual 3-dimensional structure determined by, I think, NMR, which shows you the way that this RNA is structured. And this is important for replication of the virus. So just don't look at them as just lines on a sheet. They're very complicated. All right, our last question is, what statement about viral RNA genomes is correct. A, plus single-stranded RNA genomes may be translated to make viral protein. B, double-stranded RNA genomes can be directly translated to make viral protein. C, plus single-stranded RNA virus replication cycles do not require a minus-strand intermediate. D, RNA genomes can be copied by host cell RNA-dependent RNA polymerases, or all of the above. So how did we do? So the answer is A, plus RNA genomes may be translated. Double-stranded genomes can be directly translated. No, they can't, because they're double-stranded. The ribosome can't get at the plus strand. Plus replication cycles do not require a minus strand. They do. I will show you in the slide. RNA genomes can be copied by host cell RNA polymerase. There is no host cell RNA polymerase that can copy viral genomes, right? So that's why it's just A. All right, and I end up with a few comments on genetics, how we manipulate genomes. Now, in the old days, we used to do plaque assays to do genetics of viruses. What do I mean by genetics? Well, to look at mutations in individual genes and see how they affect reproduction. So, for example, you could mutagenize a virus and do a plaque assay and pick tiny plaques and see what the mutation is and study that. But nowadays, it's totally different. We don't do that anymore. What we do is different. And before I tell you that, I want to make some definitions. A mutation is a change in DNA or RNA. So a mutation can be a nucleotide addition, deletion, or rearrangement that leads to amino acid substitutions, additions, or truncations. And as I said, in the old days, mutations were introduced by UV light, mutagenesis, chemicals, and so forth. But today, we have DNA copies of viral genomes that we mutagenize to make specific mutants. So we have the viral genome has a DNA copy in a bacterial plasmid. Now, you probably know that bacterial plasmids are circles of DNA that reproduce independently in bacteria. We can harvest these and insert viral genomes into them to amplify them, and then we can make changes in the DNA. We can delete, insert, substitute, etc. And as we'll see in the last lecture of this course, we can make viral vectors to try and cure diseases of various genetic diseases, make vaccines, and cure cancers. So very powerful, this idea of having an infectious DNA clone. And we introduce these into cells by what we call transfection. And this was first shown with bacteriophage lambda, where they found the production of infectious virus after transformation of cells with viral DNA. And so transformation leading to infection, it's transfection. So there's an example. You can put RNA or DNA into cells and reproduction can occur. So this is used widely. First example was with poliovirus. For poliovirus, there's a plus-stranded RNA in it, which, if put into cells, will initiate an infectious cycle. But you can take that viral RNA and clone it in a plasmid and put that plasmid into cells, and then those plasmid transfected cells will then produce virus. You can also transcribe the DNA in vitro with a polymerase, an RNA polymerase, to make RNA and insert that into cells. By the way, this is how the mRNA vaccines were made. They had the spike protein coding sequence in a plasmid. With a promoter, they made kilograms of mRNA in vitro to make the vaccine. This has also been done with influenza virus. In this case, you need to take 8 plasmids, 1 for each segment of the genome, and each segment makes both plus and minus strand RNA. You introduce all 8 into cells, and out comes influenza virus. And this was used to recover the 1918 influenza virus. So if you remember, you don't remember, no, you may know, I don't even remember. You may have heard or read that in 1918 there was a pandemic, the biggest one, killed many, many millions of people, but we didn't have virus until 1933, and it was no longer the 1918 virus. But investigators recovered viral RNA from autopsies and from buried victims. They reconstituted the whole sequence, made 8 plasmids, put them in cells, and out came the 1918 influenza virus. And I did an interview with Jeff Taubenberger, who was one of the main authors of that study. Very, very interesting work. It took 10 years to reconstitute this genome. Now, I've told you that you need to have a plasmid and put the viral DNA in it. By the way, if you have an RNA virus, you simply make a DNA copy of it and put it in the plasmid. Same result. Here's an example where investigators wanted to recover horsepox virus. So this was a pox virus of horses which had been extinct. But we did have the genome sequence. So investigators synthesized 10 overlapping DNAs at a cost of $150,000. They took these 10 DNAs and threw them into cells, and out came horsepox virus. They recombined very efficiently and made virus. And they had some markers inserted into these DNAs, restriction sites and yellow fluorescent protein, to show that it was synthetic virus. So this experiment and the flu experiment got people very upset. Here, for example, scientists is a headline article. Scientists bring back extinct horsepox virus in the lab, raising important biosecurity questions. And so the US made a committee to provide advice, guidance, and leadership. These are experiments where you could make horsepox virus for good reason, but maybe it has a bad use also, which we call dual use. And so this committee helps to mitigate any issues that might arise from that. Of course, there are always the naysayers who say we shouldn't be doing any of this. An example is gain-of-function research. Now, having infectious DNA copies of viral genomes allows you to do gain-of-function research very easily, right? You could do it in the old days, right, before recombinant DNA, by simply chemically mutagenizing, passing a virus in a different host. But the goal was always just to give a new property to an organism, any organism. And today we can modify infectious DNA and get different phenotypes, and that's a gain of function. It's not necessarily bad. But here are 2 examples of gain of function research. On the left, this is a 1939 paper where they took a poliovirus and passaged it in rodents and got the virus to infect rodents. So this is a virus that only infects humans pretty much. They could adapt it to a new host by multiple passages, and now that's a gain of function experiment. On the right is the kind of experiment that got the EcoHealth Alliance and the Wuhan Institute of Virology in huge trouble. And this is work that they were doing. They would take coronaviruses that infect bats and substitute in spike protein genes from other wild bat-infecting coronaviruses and see if they change their properties. So here we're looking at weight loss as a measure of pathogenicity. And you can see this green virus, the virus caused greater weight loss in animals. So that's a gain of function. It's more pathogenic, but some had no changes whatsoever. And so this is the famous experiment where Rand Paul in Congress said to Tony Fauci, "Don't you admit that you were doing gain of function in Wuhan?" And unfortunately, Fauci said we were not, which was wrong. They were, but it's not the kind of gain of function that would harm anyone. So Fauci's definition of gain of function was the old ways, passing virus, chemical mutagenesis. He doesn't consider modification of DNA to be a gain of function. So now there are all kinds of political implications of gain of function. Risky gain of function studies need stricter guidance. Fight over COVID's origins renews debate on risks of lab work. So, as I said the other day, the congressional report says the greatest evidence that the virus came from a lab is that they were doing gain of function research, which, it's just an anti-science statement. It makes no sense whatsoever. NIH documents provide new evidence that we funded gain of function research in Wuhan. That's the experiment I just showed you. There's no secret there, but it's nothing. It's not even a human coronavirus. I don't know why it matters. Republicans spin NIH letter about corona gain of function. So now you're going to see, with the new administration, restriction on certain kinds of gain of function research. And since we're going to have non-scientists running all of these science institutes, there's little that we can do to argue logic. It's really going to be a very difficult time. Okay, but on a lighter note, next time we're going to talk about how to build viruses.